picking up with anemia. This can be another pretty heavy concept. It's not as simple as it sounds, so we're going to dive into it. So this is interesting, and I had a prior, I misspoke on one of my other videos. I was thinking it was 250,000 red blood cells a day, and I was not even close to remembering the true value in healthy individuals. Just take this in for a second. 2 million a second. I can't even like physiologically imagine 2 million red blood cells. That's crazy. And that's not me making this up. This is from the article Prognostic Significance of Using the RDW. Brilliant article, brilliant article. I have a feeling you either hate this stuff or you're totally fascinated by it. If this is something you want to do and you think that reading labs and diagnosing and analyzing people's biochemical breakdowns along the lines of how I do to restore health in the population of people who aren't getting any answers by the rest of the medical community, then I would read this article, because it's a good one. Okay, 2 million red blood cells per second. Why is that important when it comes to analyzing anemia? Well, it kind of puts it into perspective, right? So what do you think the bone marrow is doing when it's not making 2 million? Or what about the poor bone marrow that's desperately trying to keep up with 2 million precursor cells a second, right? And it's like, I don't have B9, I don't have B12. What do you want me to do? I can't make heme. So kind of just goes, yeah, all right, well, maybe I'll give you a pass. It just puts macrocytosis and microcytosis like into perspective. So I'm going to let you know that I do not expect you to know all of this, and you do not have to memorize the names of the different cells except reticulocyte. And of course, by now, I hope you know that a mature red blood cell is called a erythrocyte. So that should not be too much of a problem. So this is a wonderful chart. If you want to read all of that, you can. However, here's what I want you to know. So when we're talking about macro and microcytosis, right, the ability of the bone marrow to synthesize progenitor cells that are going to become red blood cells, whether it can or cannot make normal size cells, comes back to the sequence of events of how synthesis of a red blood cell happens. So in all simplicity, since the very first stimulation of the red blood cell, a stem cell, the one that's like the mother daddy of them all, right? This is where they all come from, this guy right here. So from the moment that gets stimulated, you have to automatically have B9 and B12 to synthesize DNA. So we know that DNA, both purine and purines and, okay, of course, I just blinked on it. The two forms of nucleic acid that make our four bases, right? They require B9 and B12, and both B9 and both B12 are important for the synthesis of DNA, and that starts from the very start of the stem cell being stimulated. Once the stem cell starts to divide, that's when we start making the hemoglobin. But remember, you can't make hemoglobin if you can't make heme, and you can't make heme if you don't have succinyl-CoA and glycine. And at the very final stage, then you incorporate the iron. So from the very get-go, you better have B9 and B12. In the very early stages, the hemoglobin is starting to be synthesized and packed. Remember, in healthy individuals and healthy bone marrow, the hemoglobin is going to be maxed out at 34% of the total volume of the red blood cell. That's normal homeostasis, that's healthy function. As it continues to grow, then we're going to see here that the DNA stops, RNA continues and then slows down, but the hemoglobin still keeps getting made. It still gets getting packed in there, because what's going to happen is the DNA nuclear content is going to become pushed up against the wall and then eventually kicked out. So that's going to be the important part of how DNA nuclear content is going to be correlated to the amount of hemoglobin that can get packed into the normal sized red blood cell. And then that's an indicator as the hemoglobin gets maximized, then the nuclear content kind of gets pushed to the wall, and then it gets extruded. It literally gets kicked out. From that point on, then we're going to basically have that reticulocyte. And that's what I want you to know is the immature red blood cell that enters circulation. It only stays a reticulocyte about 2 to 3 days. And what happens in that stage of the reticulocyte is the rest of the cellular content is basically being farted out of the red blood cell. Everything gets kicked out except the hemoglobin that's in there, and the only two pathways that remain, the only two metabolically active pathways in the mature erythrocyte is going to be glycolysis, because remember, that's the only way that the cell has any ATP being made, and that's going to be dependent on glucose uptake for glycolysis generation of ATP to keep itself powering. And then the other glucose uptake pathway, which is your pentose shunt, and the very important molecule, you need to know this, is NADPH. So the two things that the red blood cell does metabolically in its mature stage in that 120 days that it's in circulation is glycolysis to stay alive, and then pentose shunt, still using glucose, just in a different pathway, and that is to synthesize NADPH. So NADPH is the molecule that makes sure that we recycle glutathione. So we're going to see that the red blood cell is super susceptible to oxidative stress and damage, and we need sufficient amounts of the NADPH being synthesized, because that keeps the glutathione levels high, which allows the red blood cell to fight off oxidative stress. So we need B9 and B12 for DNA synthesis from the get-go. Then hemoglobin and the bioavailability of iron and heme synthesis is 2/3 of the life cycle to make it to the red blood cell mature reticulocyte being kicked out into the the circulation in order to become the erythrocyte, which is the actually mature red blood cell. So immature is reticulocyte. It only is itself for 2 to 3 days until all of the other cellular contents have been extruded. I think I just picture like a little farting red blood cell as it gets rid of everything. It's just my brain. And then it becomes the mature erythrocyte, which only has glycolysis and pentose shunt for NADPH to keep its glutathione recycled. So that's the simplicity way that I want you to start rethinking about why anemia is important. And even before anemia, if we need this to be happening at 2 million cells per second, this is happening in each one of them. Each one of them has to have all of this going on. That's really important. So another critical concept, and if you read enough medical articles and textbooks then on anemia and macrocytic and microcytic pathological processes towards anemia, you're going to realize that when it comes to the word anemia, and what anemia means is nothing more than not enough red blood cells in circulation. So if you are actually anemic, which means you don't have enough red blood cells in circulation, that is not a diagnosis. It's nothing but a presentation of an underlying condition that needs to be addressed. That's why I focus so much on macrocytosis and microcytosis, because that pathological process is way more important and happening long before your bone marrow can't make enough red blood cells. So that's why we harped on it. I want you to know that anemia is not a diagnosis. It's a presentation. However, this does not mean that you are not putting the diagnostic ICD-10 code of anemia properly onto your SOAP note. This means that in the realm of what's going on with the body, finding of anemia, not enough circulating red blood cells, does not satisfy what's really going wrong with the body. So be sure you understand that. And then again, anemia is not enough red blood cells in circulation. So what we are adding now to our CBC analysis is now this is RBC less than 4. That is what gives us the diagnosis of with anemia. Because if I give you an indicator of microcytosis and I ask you for the diagnosis and you do not have an RBC count less than 4, you better not call that macrocytic or microcytic anemia. That's microcytosis or macrocytosis. Once you get that less than 4, then you can change the wording and you change your diagnosis to microcytic anemia, macrocytic anemia. Very big difference. Make sure you don't get those exam questions wrong. So what's happening here? Remember the picture of the 5 biological breakdowns before you get to end organ failure or onset of disease, right? It was stage 3 of 5 really before the anemia really kicked in, right, on the other picture. So morphological changes, that's a biochemical SOS, right? That is like, help me now, right? Fix me, fix it before it gets worse. That is an SOS warning. The very second that you have an MCV, even shifting to the end ranges of the abnormal, intervene. Catch it before your RBC drops. If you put two CBCs together, and you compare a patient that provided a blood work, lab work that was maybe 6 months ago or a year before, and you put it in an Excel spreadsheet and you're watching the pathological pattern happen in front of your eyes, if they were at a 5.1 RBC and they were like, 92, 93 MCV, and they're starting to not feel good, right? They can't really describe it. It's non-generalized. They don't really know what's going on. And 6 months or 9 months or a year later, you do another follow-up blood work and you put it in, and they went from that 5.2 down to 4.3. Is 4.3 wrong? No. What would we call that? We would call that within normal limits. But do you think that they should crash all the way from a 5.1 red blood cell in circulation 6-9 months later to now 4.2? What's going to happen if you don't intervene? It's going to continue to drop. So the pattern of rising and dropping is just as important. So we want to catch them here, right here. Aberrant MCV and the dropping RBC is what we're paying attention to. Again, that's the biggest indicator that we're going from an insufficiency to an outright deficiency. And the lab work screams these signals at us. This is when we, as brilliant, intelligent, analytical doctors, go, I know what's wrong. When nobody else out there is. I challenge you. I get goosebumps. I challenge you. Be better than what's out there. It's not that they're bad. They're just not trained to be analytical diagnostic doctors. They're trained to be puppets. Oh my gosh, I didn't mean to say that on video. Please don't play that again. So they're just not educated in the same manner. So please take advantage of the amount of knowledge and ability to change people's lives, because one day this could be your blood work. All right, anemia is then the end stage. That was trigger not removed, SOS signal not heard, right? Doesn't mean you can't fix it. It just means you're gonna have to work a lot harder at it, and you've probably got a lot more downstream negative effects. All right, so by now, you better be comfortable with RBC, MCV, RDW. You know your ranges. You know the underlying drivers of an abnormal MCV. And now we're adding on anemia with an RBC less than 4. Crap. Now we have anemia too. Now what? Now we ask the questions. Why did this happen? Okay, so MCV, we looked at the reasons of why we would have too big of cells, why we would have too small of cells. Now that we added in another abnormal finding, we're adding in that extra breakdown of biochemical physiological function and total derangement of an RBC count that is not sufficient to take enough red blood cells to deliver oxygen. right? To carry away carbon dioxide, to help fight tissue acidity. So this is, RBC is too low. That's all this is. Too few of RBCs in circulation. That does not tell you anything about the underlying cause. All it tells you is too few of red blood cells in circulation. So now you ask why. Why do we have too few? Here's the ones that we're going to look at. Okay, these are the 4 primary reasons that we have to diagnostically, analytically assess in the patient sitting in front of us with an RBC too low. And they're going to be chronic blood loss, bone marrow insufficiency, spleen. Is it acting too much, right? We're going to see cases where the spleen's like, man, well, you know, I'm just doing my job. It's not my fault you don't look right. Okay, so we're going to see what that means. And then intravascular. So what we're going to see is these two are addressing hemolysis. Hemo, blood, lysis, bursting. Hemolytic anemia is a very important concept of what we have to look at when we also take into account some of the nutritional deficiencies and the potential of extra blood loss. So let's jump into it. All right, we're going to start with excessive blood loss. The first thing you have to think of with excessive blood loss with anemia is iron deficient anemia. What are you losing with a blood loss? You're losing the whole thing. You're literally losing red blood cells. So it might not be that your bone marrow doesn't have the substrates and the essential nutrients. That 200 million a second probably can't keep up with that excessive blood loss. So right away, when you have the RBC anemia, you're most likely, well, of course, this would be microcytic. However, it doesn't have to be, because if you also have B9 or B12, that the aberrant size red blood cells could be competing. Because if you have some B9, B12 insufficiency, some red blood cells are going to be macrocytic. And as you get more and more iron deficient, some red blood cells are going to be iron deficient. And then when you have those competing processes, guess what? You might have a normal size cell. I won't test you on that. Don't worry. I'm just letting you know it's really complicated. All right. So excessive blood loss, I want you to immediately think IDA. IDA is iron deficient anemia. So typically chronic blood loss. So we have to find, why are we bleeding? Okay. So this is important because right here, if you are losing more red blood cells than you can make, what happens in the loss of red blood cells compared to splenic breakdown, we lose the ability to recapture the heme that is inside all of those red blood cells. So when we lose them to the exterior, such as blood loss, there's no recycling, there's less unconjugated bilirubin, and you are virtually immediately going into an iron deficiency because we're not recycling that pool of hemoglobin being broken down into heme being broken down into the constituents that are going to go into the unconjugated bilirubin. So as heme is broken down, it goes through several steps, and then it becomes unconjugated bilirubin. So that is a net loss of iron. And then, of course, as the iron deficiency gets worse, then the erythropoiesis is going to get worse because the red blood cells just can't keep up. So what are the primary reasons, right? So the two big ones that you really need to think of are going to be menorrhagia in your menstruating females, and then the internal bleeds. Now, without stereotyping, as patients and people age, they become more and more susceptible to internal bleeds. So if you have a history of chronic NSAID use and chronic aspirin, you are more susceptible to gastric bleeds. And then, of course, there is always your peptic ulcers, even though our ulcers are kind of getting less and less, but we have a lot of hypochlorhydria, and that can be an issue. The, the gastric bleeds are, of course, going to be almost all of the literature says if you have an older retiree or elderly patient and they show up with microcytic anemia, it is an internal bleed until proven otherwise. I'm going to say that again. Postmenopausal, retiree age, and elderly populations with the iron deficient anemia, it is an internal bleed until proven otherwise. And your menstruating females, you have to be asking all of those uncomfortable questions. How long is your cycle? If your cycle is less than 28 days, how long has it been short? I know a patient who was having her period every 17 to 18 days, and that was with 2 to 3 days of clotting. She could not sleep through the night. She was sleeping on a garbage bag because she kept staining her bed. You think she's iron deficient? Yeah, she looked like a walking ghost. Poor thing. It happened really fast too. So, is iron deficient anemia the diagnosis, or is it the menorrhagia causing the iron deficient anemia? Point being, finding iron deficient anemia is not a diagnosis. Now you got to find does why does this patient have the IDA? So how long is your cycle? How many days do you clot? And the way to quantify that is how many super tampons do you go through an hour, and how many days of that do you have? So this is a really big problem. I had a young female patient that was, I think she was like 19 or 20 when I started working with her. She had her period since she was 12 years old. Her onset of menstruation at 12, that's young. That's a little girl, right? Her menstruation at the onset was already with cramping, severe pain. She had to stay home from school all the time. Her normal from her very first period was clotting, pain, and severe heavy menstruation. And do you know that nobody talked to her in like 6 to 7 years to say that that's not what a normal period is supposed to be like? She's probably been iron deficient since she was 13. So yeah, iron deficient anemia is not the diagnosis, it's the presentation. It's the screaming SOS signal that something else is wrong. So you quantify a female menstruation heavy clotting with how many super tampons are you soaking an hour, and can you make it through the night? Those are the clinical questions that I want you thinking about when I test you, okay? So when we're thinking about an internal bleed, the primary places are gonna be GI, urinary, and genital. So if it's going to be a GI bleed, you've got to be thinking like the separation of the desmosomes, that's a gastric bleed, or you have the peptic or duodenal ulcers. Those can go slow, insidious, completely undetected for years. If it's a slow bleed, it's not even enough blood to enter into the intestines for feces to even change the formation of the stool. It might not be tarry black stools like we learn in pathology. It can be so subtle that they're just losing chronic blood slowly all day long, right? That's why it's even scarier. At least a menstruating female knows they're going to the bathroom with clotting. A person who has a slow, insidious gastric bleed doesn't know, and it can go on for years. So peptic ulcer and gastric bleeds, cancer is very prominent to be a slow bleed into the intestines. That's why it's really important to rule out GI bleeds because of their insidiousness. We know that IBD is correlated with the ulcerative colitis. And then, of course, parasites, undetected or undiagnosed, can also cause slow gastric bleeds. Urinary, you've got to be thinking about chronic UTIs. You would be testing for positive white blood cell esterase, potentially if it's going to be correlated with an infection. And then chronic stones, as well as BPH or other common sources of slow urinary blood loss. And then kidney cancer can also be, or even bladder can have a slow bleed that can go undetected if it's not enough to be like bloody in the urine. And then, of course, genital, we want to go back to any of the menstruation or any of the cervical or vaginal blood losses, and then your cancers. So, already said that. Make sure you understand that it's an internal bleed until proven otherwise. Here's a really cool picture. This confirms what we already learned. This process goes on long before you finally show up with iron deficient anemia. You do not have to memorize this graph. You have to appreciate the concept that you are going to show with aberrant changes in shape and iron stores, ferritin, circulating iron, long before you finally end up with your outright anemia. So this is days, months, and even years, and that's pretty much the big take-home message. All right, next concept. So now we're looking, instead of chronic blood loss, now we're moving on to another reason, being bone marrow, anemia of diminished erythropoiesis. This is where we're looking at our nutritional deficiencies. So the anemia of inadequate production of RBCs stems from the inability to make red blood cells because we don't have the nutritional essential precursors. This is straight from Robbins, and even Robbins Pathology says one of the most common and most important causes of anemia is that chronic, persistent, unfixed nutrient deficiency, as we've already pounded into our brains for B9, B12, iron, and heme. So what we need to know now is that we are taking it to the next level. If you have macrocytic anemia correlated to B9 and B12 inability to synthesize DNA, that is now the proper diagnosis of megaloblastic. Megaloblasts are red blood cells that are too big because they have too little and underdeveloped DNA. Okay, so that's very important. Then we also have the iron deficient anemia, as we just looked at. So if you have outright this iron deficiency, then that is not enough precursors for red blood cells. Iron sequestration, this is a new one that we're going to introduce. When you have chronic disease and chronic infection or inflammation, the body hides the iron, and we're going to learn that. And then the last two that can also cause a problem with bone marrow are aplastic anemias and then your neoplastic anemias, and we'll take a look at those. So this is where you're going to get more exhausted, more details with methionine synthase and the methionine cycle, and it's that important because we are learning the association of hyperhomocysteine, and when we start to look at atherosclerosis and coronary heart disease and all of the negative effects that happens in the blood vessels, it also comes back to this pathway. So we're going to jump around. I hope by now, I'm not going to read this to you. You need to know every detail on every bullet of this slide. Super, double, triple star. This is a large fraction of being able to diagnose the CBC and the RBC and the MCV, and this is going to come back to haunt you when we look at the atherosclerosis and we're analyzing the CMP. This is a really important slide. So double star, triple it. Now, what I'm going to do is show you some really cool pictures. Okay, so I'm going to jump into another slide here. Yeah, come on, give me the PowerPoint. It's very delicate mouse maneuvering here. Okay, so I uploaded an extra PowerPoint. I don't think I screen shared, so let me screen share. Which one is it? All right, now you should be able to see it. So this is going to be a PowerPoint that I uploaded as an extra, there's extra pictures. So this right here is the folate cycle. And what we need to focus on here is the 5,10-methylene, right? That's the molecule that can go either this way into the methionine cycle, or it can go down this way. So the way that you need to read this, 5,10-methylene tetrahydrofolate, is directly follow this loop, boop, 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 all the way up there, right? So the 5,10-methylene directly is a cofactor in the nucleic synthesis of the thymidine. So that's our thymidine. However, what's important to understand right here is as 5,10-methylene tetrahydrofolate makes thymidine for the nucleic acid, for DNA, it also regenerates dihydrofolate. That's called recycling the folate pool. In order for folate to be in all of these different forms, they all have to, they transfer back and forth. So as 5,10-methylene makes 5,10-methylene tetrahydrofolate makes thymidine nucleic acid. It also regenerates your dihydrofolate as part of the folate pool to then become tetrahydrofolate again through an extra step, which is now the active form that the body uses and puts tetrahydrofolate through all of those different derivatives. Now, we also have the same molecule, 5,10-methylene right here, tetrahydrofolate. This is your irreversible reaction, MTHFR, that's going to be an irreversible reaction that locks folate into the methyl B9. Methyl B9 is only used in the methionine synthase, and that's where the methyl group is passed off to B12 in the methionine synthase enzyme, which then passes that off again onto homocysteine, preventing hyperhomocysteine while simultaneously regenerating methionine, which then immediately converts back into the SAMe acetylenolmethionine as we cycle methionine right here. So we are making sure that the folate pool is being sustained because folate in its different active forms is used in all of these. And as the methyl B9 gives the methyl group to B12 to homocysteine to regenerate methionine, it also, just as importantly, as the methyl comes off, the molecule itself, just the basic tetrahydrofolate, is regenerated. And that's a key feature because that supplies the folate pool. That tetrahydrofolate right here immediately goes through another conversion to remake the 5,10-methylene so that it's again bioavailable for thymidine. So that recycling is very important. Right, I'm going to switch screens again. So let me show you this in a better picture. This is from Groper. So this is what we were just looking at, okay? So here's folate. Folate is what we get when we eat the natural food sources that contain the folate from the leafy greens. If you have a supplement or a fortified food, which is folic acid, then that has to go through this dihydrofolate in order to be synthesized and converted into tetrahydrofolate. So this is where everything goes here. And remember, the active form is tetrahydrofolate. So I'm going to go big. Let's look at this in a little bit more detail. So if we look at tetrahydrofolate, it has to go through the conversion into 5,10-methylene. 5,10-methylene unidirectionally makes 5-methyl. We call that methyl folate. And then this is a one-way irreversible reaction. This is the one that passes the methyl off of itself onto the B12 dependent enzyme, recycling homocysteine and regenerating methionine. But while it does it, it regenerates tetrahydrofolate for the folate pool. If we have low B12 and we have a methyl trap, that means that methyl B9 is trapped in this irreversible direction. That means that we are not regenerating enough tetrahydrofolate for the folate pool. That directly causes megaloblastic macrocytic anemia because there's not enough tetrahydrofolate to be put back into the 5,10-methylene, which goes boop, 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 follows all the way up here. And then we have our direct purine pyrimidine. That's what it is, pyrimidine. Yes, synthesis, and that's your thymidine pathway, while that reaction regenerates the dihydrofolate so that it can go back into the tetrahydrofolate. Don't get lost in it. The concept is replacing the folate pool with the different steps and the shared pathway of the 5,10-methylene to make our available nucleic acid, as well as to make sure that we are getting the methyl folate into the pathway. So I will show this to you in another format if you wanted to see it again, because I think it's valuable to just, picture speaks 1,000 words. You can hear me say it, you can read it, but there are different ways to see how they're showing the integration and interconnectivity of how these all balance out. So this is the Linus Pauling, which is the Oregon State for micronutrients, and this is folate. So if you scroll a little bit down onto their page, they do a beautiful job of drawing. Here is the B9 and B12 interconnection. So folate, as the 5,10-methylene, goes into nucleic acid synthesis, but it also goes into the 5-methyl, which is the methionine pathway. And then they're showing here that as B12 takes the methyl group off, we are regenerating that tetrahydrofolate. And then it's right here. If that doesn't happen, this is why B12 is also indirectly correlated in megaloblastic macrocytic anemia, which is the ultimate inability to make red blood cells sufficient with DNA. Megaloblastic anemia is B9 and B12 deficiencies and underdeveloped DNA. Underdeveloped DNA is megaloblastic macrocytic anemia, and it's all right here because we're not generating the folate pool to allow the folate to go back, the tetrahydrofolate, to go back into the form 5,10-methylene to make the nucleic acids. So I hope that clarifies as much of the cycle, because you probably never want to hear it again, but I hope that's a really great indicator that we are learning it. So that was this slide in a nutshell, and those are the resources if you want to take a look at it. So we've looked at this one before. Okay, let's look at it from another perspective. Number one. Oops, that's not what I wanted. Get you out of my way here. Okay, so we have the physiological finding of megaloblastic macrocytic anemia. Megaloblastic macrocytic mean not enough DNA, too little B9 and B12, and underdeveloped nuclear material in the red blood cell. So the resulting pathological, pathophysiological effect is impaired methionine cycle. Make sure you understand that. Impaired methionine cycle due to low B9 or B12, right? That causes the formation of the megaloblastic impaired DNA underdeveloped macrocytic red blood cell. Make sure that you understand how that all comes in together. So now that we know that we have an underdeveloped DNA megaloblastic macrocytic red blood cell, we know that that immediately also causes elevated homocysteine, which is your cardiovascular risk for clot and thrombus, and that is a direct driver of atherosclerosis. Simultaneously, that same process also means that we now have decreased SAMe, because we have insufficient recycling of homocysteine, inefficient regeneration of methionine, and now that causes less bioavailable generation of the universal methyl donor S-adenylmethionine. That is an epigenetic risk for cancer, as well as lots of disease, aging, degeneration, epigenetic negative control, and that can also correlate to mood, anxiety, and depression. Here is one more picture. I like this one because it correlates how 5,10-methylene is involved in that pyrimidine for that thymidine synthesis, as well as that 5-methyl. Here's another picture. Again, pictures speak 1,000 words. We are almost done with this, I promise. The methionine synthase is your methyl transferring the methyl group onto B12 while regenerating tetrahydrofolate. Then the tetrahydrofolate is now available as your folate pool to become the form 5,10-methylene, which is the form necessary to make your thymidine. I hope by now we have driven home all of that so that it's becoming more comprehensible, and it's becoming less, you don't have to think of it so fast. I'm drawing a blank on this word today. All right, causes of megaloblastic macrocytic anemia. This is everything that we covered in the prior lecture. So what do we need to be thinking about? Are you not eating enough of it, low intake? Are you not absorbing it? We already talked about the causes, so make sure you know all of this, know this well. Are you losing it? Or do you have an inherited SNP? Is it a polymorphism that's changing the efficiency of your enzyme function? Now, what does this mean clinically? What are our signs and symptoms? If you have megaloblastic macrocytic anemia, you have all of the symptoms already associated with macrocytosis. You've just got anemia on top of it, but that means that you really don't feel good, right? You're unwell, you're fatigued, you're exercise intolerant, you're likely to be shortness of breath because you're not delivering enough of your red blood cells. You're not healing well, and you also have, especially if it's a B12, I want you to think of a neurologic. That's your paresthesias, that's your piriformis and sciatic-like presentation, and also once B12 gets really bad and the myelin starts to significantly be impaired, then you're going to start to have the vibration sense, proprioception decrease, like you're going to fail your Romberg's test. So think neurologic for signs and symptoms. This is the LabCorp. If you type in anemia and you search the tests, there are different panels. There's a panel A, a panel B. I won't ask you to, I'm not going to test you on knowing the panels. What I'm showing you this as a clinical pearl to know that there are different types of tests that you can do. And then the megaloblastic panels here, panel A versus panel B. What I want you to see here is the testing. So panel A includes your CBC with differential, always get differential. Here's your iron binding capacity, and your reticulocyte count. That's valuable. And then your iron B, anemia panel B, is your CBC with differential. This has your ferritin, your folate, iron binding, also your reticulocyte and your vitamin B. Now, none of those are your functional markers for B9 or B12, so check this out. A much more sensitive marker of megaloblastic correlating to your B9 pathway is this methylmalonate. Methylmalonate is your functional B12 marker. Know that. Methylmalonate is your functional B12 marker. It's the most sensitive indicator of a B12 deficiency long before B12 goes deficient in the serum, long before anemia develops, so be sure you know methylmalonate. However, this is cool, right? If you remember the transsulfuration pathway where homocysteine goes down into cysteine, it requires 2 steps of B6. Well, that's going to be your cystathione and your homocysteine right here. So if you did this panel, and I will let you know this is $159. I looked into it. I was going to get it for myself, but if I go through a doctor, which is a waste of my time because they don't know how to read lab work, then I could get it free. But then they have to order it, and a lot of times I can do it much faster and I'm much more efficient. So just have to decide if I want to drop 149, 159 bucks. But it gives you homocysteine and methylmalonate all right there. However, notice though that it did not give you the CBC. You have to order the CBC separately. I can get a CBC for $12. It's very valuable. All right, these two talk about the time span. I'm not going to read that to you. Be sure you read it. Here's another picture. I'm sure the last thing you want is me talking about the methionine cycle again. Whew. All right, switching gears. We are now going to talk about iron again. Whew, let me catch my breath. It's exhausting, isn't it? I hope you know how to draw out the homocysteine pathway. I am challenging you to please make me proud. Make me proud. Go forth into clinic and know homocysteine and your methionine cycle. Okay, why else would the bone marrow have diminished erythropoiesis? Remember, we're going to come back here and look at the reasons, right? So we just looked at megaloblastic. Now we're going to analyze these two together. Iron deficiency anemia, we don't really have to talk about that. We already talked about it up here. So if you want to review it up here. I'm going to pass on that quite fast, and we're going to jump right into iron sequestration. And then aplastic and neoplastic are super simple. There's virtually nothing to learn about it. So that's like a spit's worth of breath, and we'll talk about that in a second. So let's take a look at iron deficient anemia. Right? So what are we really looking at if we have diminished erythropoiesis correlating to low iron and low heme? The two reasons are iron deficient anemia or anemia of chronic disease. That's the next thing you really need to plant in your brain. If you have a patient with microcytosis and anemia, microcytic anemia. Okay, iron bioavailability. So here's the three things that you need to know. Are you not eating it? Are you not absorbing it? Piece of cake. Are you bleeding and you're losing your iron? Duh. Here's the new one. Iron sequestration. Oh, that sounds kind of cool. What the hell does that mean? Iron sequestration means that your body has iron, but it's locked away. Okay, so let's see what that looks like, okay? If we have iron deficiency anemia, we have insufficient bone marrow erythropoiesis, we're going to have less than 4, that's anemia, and of course microcytosis, less than 82. All right, you better remember that you should be ordering both an iron panel and ferritin. I hope you know by now that the ferritin does not come standardized. I've said this a couple times, we've looked at it. An iron panel does not come standardized with ferritin. You have to order ferritin. Know to order ferritin. Did I say it? You've got to order ferritin. It's not standardized on an iron panel. However, it's the most sensitive indicator of your iron stores, and it's going to tell you that there's a problem long before your serum iron goes down and long before your bone marrow crashes. SOS, SOS signal, SOS, right? Get it before it's a problem. We've already talked about this. I'm not going over it again. Refer up higher into the PowerPoint or rewind. Now, maybe I need to say it one more time. Come to think of it. This is an important thing right here. So before I pass on it. Okay, we talked about GI bleeds, but why do we have a GI bleed? I think I said it, but let's just make sure. So NSAID history, chronic aspirin is one of the primary causes of a GI bleed. I did say aspirin, but let's make sure, because NSAIDs is another word that is commonly used in medical literature and commonly used in other offices. So you should be asking not just about aspirin, but a chronic use of NSAIDs is one of the most common causes of GI bleeds. Now, this is the next important part when we're talking about iron deficient anemia based on iron loss or chronic blood loss or insufficient food or absorption, is this right here. So if you want to think about, do you have a chronic bleed? And you've ruled out NSAID use, you've ruled out menstruation, you've ruled out digestive enzymes, you've ruled out all of the causes, and they still have microcytic anemia, you better get a GUIAC test, because that's going to find an insidious chronic bleed. So this is called a GUIAC. That's GUIAC. It's an occult stool test. Basically, it's you have to take a small stool sample. The patient, they take it home or they get it in the mail. They have a special like spatula. You catch your feces during defecation. You take a little spatula and you put it into the sample, and then the sample is going to run it for occult blood. Because again, you can have enough. You can have a low enough of blood in your stool that it doesn't become tarry, which is what they teach you, but it's not always like screaming tarry black stools. So you've got to know that GUIAC is the occult blood test, and that's what you need to do to rule out a chronic bleed that was not found inside the body, especially when we have an iron deficient anemia. So be sure you are remembering the GUIAC test. This is what it looks like. I searched GUIAC as the test because I know it's called GUIAC. However, look at this. LabCorp called it occult fecal. So be sure you recognize both, because I will test you on both of them. Okay, this is cool. This is really cool. I think it's so cool because this is what the body does. The body is really, really, really smart. The body knows what to do. If we just help it do what it's supposed to do, it will work in our favor. Now, I'm not sure if you recall from iron 2 semesters ago, however, a really interesting protein called hepcidin is synthesized from the liver in response to either high iron or chronic inflammation and or chronic infection. So what hepcidin does is twofold. Hepcidin, in response to high body iron, right? Iron, like circulating, high serum iron, because it's a very potent pro-oxidant. Because of its redox state, transferring back and forth from ferric and ferrous in the 3+ and 2+, that makes it a very potent oxidizing agent. It will go ahead and steal electrons from molecules, and that's called oxidative stress. So the body does not like to have high iron circulating free. It's a very bad oxidative process. So if the liver senses high iron in the blood, then it's going to release hepcidin. If it has chronic infection, chronic inflammation, which basically means a body habitus of chronic inflammation, how many obese metabolic syndrome patients are walking around with no oxidative stress because their metabolic dietary lifestyle has depleted all of their glutathione? Yep, like all of them. So that is a sign that the liver should be synthesizing and secreting hepcidin. Hepcidin is going to tell your intestines to stop absorbing dietary iron, and worsefold, which is how our anemia comes in, is it irreversibly locks iron into your macrophages and prevents export. That is what iron sequestration is. It is the hepcidin response of the inability of iron to be taken out of the storage macrophage sites. So this means that our body has the iron, but it's not bioavailable. It is trapped inside the cells. So what does that look like? We have a low RBC or a dropping RBC, a pattern of worsening pathological processes towards anemia, but we have low serum iron and we have high ferritin. High ferritin, low serum iron, especially in the face of anemia, we're going to call that a diagnosis of anemia of chronic disease or chronic inflammation, depending on what the person's body is. So it's a problem. Now, if somebody has an infection, the body knows that the microbe or parasite uses iron in its own metabolism. That's why this is a defense mechanism, and the body's going to take the bioavailable iron and it's just going to hide it away. You're going to get a fever, you're going to sweat out the microbe as our immune system attacks it or whatever it is. And while we have the fever, we have a temporary transient iron sequestration. However, if a person has this chronic, undealt with, low subacute inflammation and or infection, think about a person with a chronic reactivated Epstein-Barr viral infection. That's a situation I've diagnosed in a couple people, and I had one patient who outright had this perfect presentation. He went to go give blood and got kicked off the blood bus, which was really disturbing because he was a long-term donator. And I found after significant blood workup, we had a chronic viral infection. He had significant iron sequestration. His ferritin was off the charts. He was one of the lowest microcytic anemic patients I had ever seen. And it was partly due to this chronic infection that hadn't been treated. We treated the Epstein-Barr with antivirals, and then we treated the iron with iron bisglycinate, and within 4 to 6 months, he recovered and he was back on his blood bus donating blood. Because that was his primary goal. He was devastated. He didn't care that he had this walking infection and microcytic anemia, like the worst I've ever seen. He was like, I can't donate blood. So you got to find out what's important to each patient, right? So understand that iron sequestration is high ferritin. The body traps the bioavailable and makes it not bioavailable, and then it presents with anemia because it's not able to get into the tissues that need it. All right, this is everything we already looked at on the previous lecture. So why would you have low iron? You're not eating it, you're not absorbing it. Be sure you know the reasons why. Now we know to add in the concept in the additional diagnostic concept of inflammation, infection, and that hepcidin. So again, hepcidin, know it, decreased absorption and traps it in the macrophages. Do we have an increased need? Already talked about that. And are we losing it? So be sure you are thinking about where we're getting blood loss from. So what happens in our patient? What are the signs and symptoms? Be sure you know all of these. These are important. You're easily fatigued. You have exercise intolerance, very likely pale. This is a cool one, blue sclera of the eyes. That's definitely associated with iron deficiency. And then people eat non-food stuff like hair, dirt, cotton balls, and somebody who chronically chews ice is most likely iron deficient. And of course, inability to tolerate cold, guess what that is? That's hypothyroid because you don't have enough heme to synthesize thyroid hormone because of the thyroid peroxidase. All right, whew, we're almost there, right? Those are the big ones. What are the other two reasons that your bone marrow cannot synthesize enough red blood cells? Well, aplastic anemia and then neoplastic anemia are two bone marrow crashing physiological derangements. What I want you to know about aplastic anemia is very simple. It's the bone marrow benign failure, and it's not just red blood cells. That's going to show up in all hematopoietic. That's going to be all leukocytes, all your white blood cells. You're just not going to have enough of any of them. That's low, 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 low. So aplastic anemia is the inability of the bone marrow to make any of the hematopoietic. Okay, so all would be low. Neoplastic is basically bone cancer. That cancer has replaced the functional bone marrow cells, and you're simply not keeping up with the 200 million red blood cells because your cancer has eaten away at the bone marrow, and you have a nice big radiologically found cancer in there. So that's the bone marrow failed erythropoiesis. Now we're moving on to hemolytic anemias, and we are almost through anemia. We are almost through reading a CBC for the hematopoietic, and you're going to be able to like analyze all of this like really soon. Hemolysis, extravascular, intravascular. Hemolysis, intravascular, extravascular. Intravascular means that the hemolysis is happening inside the blood vessel as it travels in its journeys. Extravascular means it's not in the blood vessels, and most of the time it's going to be in the spleen. It could be in a few other places where the macrophages are. However, it's almost always going to be splenic. All right, let's see what this means because it's fairly simple. If you remember the journey of a normal red blood cell, and if you remember normal, then it's a lot easier for you to not memorize abnormal. Just think about the pathological process. Think about the why. In normal red blood cell degradation, as the red blood cells age towards that 120 days, I've taught you, they become senescent. Senescent means that they are getting older. As the senescent older red blood cell ages towards that 100 and 120 days, it shrinks itself and the hemoglobin shrinks. Both are morphological changes. Those are signals to the macrophages in the splenic sinusoids during red blood cell travels in its cycle that it should be phagocytosed and recycled. So when you think about extravascular hemolysis, what we're talking about are the reasons that cause the splenic macrophages to purposely engulf and eat the misformed, the misshaped red blood cells out of circulation. So basically, hemolytic anemia is going to be extravascular when it does not deform well. So any changes to the shape or the deformity is the ability and the flexibility of it to mold into small shapes and be flexible to get through all those small little sinusoids. They're just as small as some of the capillaries where the transport happens. So if your red blood cell has problems with its membrane and or its size, that tells the macrophages in the spleen to engulf it, eat it up, and degrade it. So that basically means that we have a short life cycle. So the extravascular hemolysis is the spleen macrophages eating deformed, shaped red blood cells and clearing them out of the circulation. So if that happens enough, then you're not going to end up with enough red blood cells in circulation, hence anemia. So extravascular hemolysis, I want you to think splenic macrophage going crazy. And that's because they're shaped wrong. They're not going through the spleen, the sinusoids well, and the macrophage goes, yo, buddy, I don't like you. I'm going to eat you. I'm clearing you from circulation. And it clears it out, goes through phagocytosis, and boom, now you have all this extra hemoglobin, unconjugated bilirubin being recycled. And guess what happens to your spleen? Splenomegaly. That's how you know you have too much unconjugated bilirubin. The spleen is too big because it's trying to take on too much deformed, malfunctioning red blood cells. And that is premature destruction because the red blood cells that aren't formed right are shaped wrong and they're being cleared from circulation. So membrane changes, deformities, not moving through the sinusoids, all of these are reasons that the splenic macrophages are inducing hemolytic anemia, okay? These are the big four. We're not going into them in detail. You've learned them in other classes. Just recognize that these are correlated with membrane-shaped problems, and that tells the macrophages to eat them, and they don't get to live 120 days because they're being cleared out of circulation. The big ones are your glucose 6-phosphate deficiency. That's the one I want you to know. That is the one that correlates to not having enough NADPH. If you don't have enough NADPH, as I had mentioned earlier in the lecture, then you don't have enough of reduced glutathione, and then your membrane becomes susceptible to oxidative stress, and then your membrane gets attacked. So that's not good. That's going to induce hemolytic anemia. So be sure you know glucose 6-phosphate as well as the ability to have enough of the NADPH to keep the normal red blood cell protected. Because even if you weren't born with a genetic mutation in your glucose 6-phosphate, glucose phosphate dehydrogenase pathway, if you don't have enough NADPH being synthesized, your red blood cell is going to get eaten by oxidative stress, and then your spleen is going to eat it by macrophage uptake and hemolysis. So NADPH, glutathione protects the membrane. That's your glucose 6 deficiency, and then hereditary spherocytosis, sickle cell, and thalassemia are the groups of membrane-changed hemolytic anemia. I don't want you to worry about anything more about that. Just know that, okay? Here are your symptoms. Of course, it's anemia. Then it's splenomegaly, as I had just said, and then your jaundice is coming from the unconjugated bilirubin because the spleen, as it breaks down these deformed, mal-shaped red blood cells, we still have all that hemoglobin in there. So the spleen is still doing its job of taking out the hemoglobin, breaking it down to the heme, and then breaking down the heme into the unconjugated bilirubin. So we are still having that, we're still recapturing at least the hemoglobin portion. However, you're going to end up with jaundice because you have high levels of that unconjugated bilirubin going to the liver. And it's the liver's job to conjugate it, and we're not worrying about that right now. So what is the difference with intravascular hemolysis? This one is super simple. First of all, make sure you understand intravascular is the red blood cell lysis is happening in the blood vessel itself during its journeys. Well, why would that happen? Very easily. The most common reason is prosthetic valves. People who have had surgeries and instead of changing their diet and their lifestyle to prevent heart attacks and strokes and decide to go remove their saphenous vein, they end up with prosthetic valves. And that valve being like synthetic, every time the red blood cell tries to like go through it, it's like so then you end up with a whole bunch of broken, damaged red blood cells, and they're breaking right there in the circulation. Another one that is damaging red blood cells as they're trying to travel through, of course, is a suprastenotic atherosclerotic vessel that's got a super big clot, because that's mechanical trauma every time it goes through. Then, of course, if you can think about mechanical, if you're like that all the time, like welding, like those guys, I picture them on like the guys that have to work like construction, they're like that all the time. They definitely have hemolytic anemia. I would love to just go blood test all of them and just like analyze all their CBCs. Like that would be like really fun. Then, of course, I think this was hilarious. This was in one of the textbooks. Your bongo drummers apparently are really susceptible to a hemolytic anemia if they over bongo drum. So make sure you don't over drum. And then, of course, the last one is the infections. Certain parasites can just eat your red blood cells. That doesn't sound very fun, but just think of it. And then if you're the unlucky person who all of a sudden has an immune system that decides to attack itself, sometimes complement proteins can attach to red blood cells, and then that causes a lysis to occur. Not good. So that covers the hemolytic anemias. So this is the difference with intravascular. Yes, you still have anemia. Yes, you still have jaundice. However, the unconjugated bilirubin is less likely because it's not being broken down. Instead, you've got hemoglobin just dumping everywhere. So you've got hemoglobinemia, that's going to be in your blood, and the hemoglobinuria, which is in your urine, it's just pouring out of your body because you're like breaking all your red blood cells in circulation. And the last but not least is going to be the add-on test. We study these in the next module. However, you need to know hemolytic anemia, whether extravascular in the spleen from membrane deformity defects, or intravascular, most likely a valve or atherosclerotic process, is going to still both cause the indirect unconjugated bilirubin as it travels to the breakdown product of the hemoglobin and heme. However, the add-on test is going to need to be the CMP, because that's going to be the measure of bilirubin, and then your UA to see if we have one of the byproducts of the unconjugated bilirubin showing up in the urine. And we learn those in detail in the next segment. However, you need to know in the hemolytic anemias, the add-on test is going to be a CMP plus a UA, which is your urine analysis. Next, we're going to take a look at a couple case studies, and we're going to look at them in entirety so that you have a patient in front of you. You've got this full CBC, and we're going to do a complete review of findings. And the next thing that we still have to do on top of that is take a look at the hemoglobin markers, because that gives us one little last piece of information that correlates to diagnosis on the CBC that will help us treat the patient. Okay, so we're gonna look at hemoglobin markers next. It's really quick, I promise.